Assalamu alaikum. Penicillium. Penicillium is a saprophytic fungus and that is commonly known as blue-green mold. Here are the contents that we will discuss: description, occurrence, general structure and reproduction in Penicillium. First of all, description. Penicillium is a saprophytic fungus that is commonly known as blue-green mold. According to the Rapper and Thom in 1948, the genus includes 136 species and the distribution is throughout the world, and they are present in the soil, air, decaying fruits and vegetables and meats, etc. And the wonder drug, the penicillin, was first discovered by Sir Alexander Fleming in London, and during his work with bacterium Staphylococcus, which was responsible for the boil and burns, etc., it was contaminated with the mold spore, and that mold spore was Penicillium notatum, and he isolated and called this antimicrobial compound called as penicillin. And later Rapper and Alexander in 1945 selected a strain of Penicillium that was more efficient than Penicillium notatum. And in the production of penicillin, the importance of penicillin is many species of penicillin are have many great importance. They can cause decay of foods, they can cause spot on the papers, books, etc., and rotting and spoilage of fruits like apple, oranges, grapes, etc. And they are also the source of antibiotic penicillin, and some are the source of and some are used in the hydrolysis of fats and flavoring of the cheese. Next is occurrence. Penicillium is a common mold and it is found on the different substrata, and it is most commonly found on the citrus and other fruits, and it is also found on the foodstuffs, jellies, old leathers and paper and in the soil. And penicillium is used for the production of certain acids like fumaric acid, citric acid, oxalic acid and gallic acid, and it is also used in the production of cheese. First antibiotic was obtained from Penicillium notatum. Third is about its general structure. So the colonies of Penicillium are bluish green, and this color bluish is due to the color of conidia, and the vegetative mycelium is hyaline, and mycelium consists of branched septate hypha, and sometimes the mycelium becomes compact to form sclerotium, and each cell has many nuclei, and the septa have perforations for the passage of cytoplasm, and the vegetative hypha grow in the substratum or on its the surface, and its reserve food material is oil globules. So what is the vegetative structure of penicillium? It is the vegetative body is mycelial, and the mycelium is profusely branched with the septate hypha that is composed of thin walled cell that containing one or more nuclei, and each septum has a central pore through which the cytoplasmic continuity is maintained, and some mycelia grow deeper into the substratum to absorb food material, and other remains on the substrate to grow on the mycelial felt, and these also have the reserve food material, and that is present in the form of oil globules. Next is reproduction in Penicillium. Reproduction in Penicillium may be vegetative, asexual. Vegetative reproduction takes place by the accidental breakage of vegetative mycelium into two or more fragments, and then each fragment grows individually like the mother mycelium. Asexual reproduction takes place by the formation of conidiospores. Asexual reproduction is by the conidia. Conidia are the globose or ovoid shape, and they have smooth surface. The color of the conidia is from green to blue, and the conidiophores are erect. They are branched like a broom, and the number of branches varies in different species, and the branches may be equal or unequal, and each branch bears at its tip and a group of short branches are called as medulla. And bottle-shaped sterigmata are also present at the tip of each medulla, and conidia are produced in chain at the tip of sterigmata. And the whole group of medulla and sterigmata on each branch is called as penicillus. And the number of sterigmata on each medulla and the number of medulla on each branch are constant for each species. So how formation of conidia takes place? Each sterigma is a uninucleate structure, and their nucleus divide into two during the production of conidia. One daughter nucleus with some cytoplasm migrates into the tip of the sterigma, and the apex of sterigma swells. A cross wall cuts off the apical portion of the sterigma to form the rest of the sterigma, and the protoplast of the separated portion contrasts, and it secretes a wall. This process is repeated several times. Thus, a chain of conidia is produced at the tip of each sterigma, and the conidia are held together by the parent wall, and these walls break and releases the conidia, and the production of conidia is endogenous. Each newly formed conidium is uninucleate, but later on this nucleus divide and it makes the conidium multinucleated, and then the conidia will fall on a suitable substratum and will produce a hypha directly, and then conidia will give the bluish green color to the sexual reproduction. Sex organs are developed on the tips of the certain hypha. Penicillium may be homothallic or heterothallic, means sometimes its thalli can be similar or its thalli can be different. First of all, escogonium. Escogonium is the female sex organ, and it has an elongated club-shaped structure, and it is uninucleated in the beginning, but its nucleus divides and it becomes multinucleate. Second is antheridium. The male sex organ is called as antheridium, and a nucleus migrates to the tip of the antheridial filament, and this tip is cut off by the septa and antheridium is formed, and the antheridial filament coils around the escogonium. A coiling will be formed. Okay, so the antheridium will come in contact with the escogonium wall, and the wall dissolves at the point of contact. Thus a passage is formed between the two, and the protoplast of the antheridium migrates into the escogonium, and then plasmogamy may occur, and then clastothecium formation will occur. So the mature escogonium will divide into the number of binucleated cells, and each cells give rise to one or more escogenous hypha. And sterile hypha will grow from the mycelium, and it encloses the sex organ to form the peridium, and thus a characteristic rounded fruit body clastothecium is formed. Then ascus and ascocarp will form. How ascus and ascospores will form? Nuclear fusion will take place in the terminal hypha that will give rise to globose asci, and ascogenous hypha directly functions as an ascus mother cell, and then fusion nucleus divides thrice, and first division is meiotic. Thus 8 nuclei are formed, and each nuclei will changes into the ascospores, and the asci are irregularly scattered within the clastothecium, and the ascus has 8 ascospores, and the ascus wall breaks and releases the ascospores within the fruiting body, and the ascospores are smooth. Sometimes they have various types of ornamentations on the wall, and the wall of clastothecium breaks and releases the ascospores. So look at here. Here is the thalli, and then it is the young escogonium, and here what happened? Escogonium is the female sex organ, and this male antheridium will coil around itself, and then after coiling they will form clastothecium, and then ascus formation, and then ascospores will be released out of the body. Now look at the life cycle. In the life cycle, conidiospore conidia are produced, and then the chain of conidia will produce many conidia, and then they will germinate into the spore, and the germinating tube will form, and then they will form a vegetative mycelium. And now the vegetative mycelium will have the young escogonium, and on that escogonium there will be two thallus that may be heterothallic or homothallic, and one will be the escogonium, and the coiled structure will be the antheridium. And around this escogonium, this antheridium will coil around the escogonium, and then antheridium comes in contact with the escogonium wall, and then the wall dissolves at the point of contact. Wherever the coiling is happening, the wall will dissolve, and thus a passage will form between these two thalli, and then plasmogamy will occur. And then terminal binucleate cell will form, and then also some bud will arise, and then ascus mother cell will form, and young ascus will form the ascus and ascospores that will form the body of clastothecium. And on the clastothecium, many spores will form. First the mitosis will occur, and then 8 nuclei will form, and each nuclei changes into the ascospores inside this clastothecium, and then ascus wall will break and will release these ascospores within the fruiting body, and then ascospores will be of the various shape, and they will release out of this clastothecium, and then they will germinate into to form a new vegetative mycelium. If you have any question regarding this, you can ask me, and thank you.